everyone, warm welcome to our channel. Today we are discussing about the Vibrio cholerae. It is our second class of the basic microbiology. See here, we have already covered the Escherichia coli. Today we're going to discuss about the Vibrio cholerae. Just subscribe our channel for daily classes as well as just check our description box for all kinds of update related to M.L.T. See, before starting the Vibrio cholerae, I just want to say that I have already told you that how to you study any kinds of bacteria. Whenever we are discussing about any kinds of bacteria, we need to take the introduction part at first, after that morphology, after that culture characteristics, after that disease condition, and after that lab diagnosis, after that treatment portion we need to take or we need to know, right? I have already told you that how to you study any kinds of microbiological topic. See, here is a basic introduction, that is a gram negative bacteria. I have already told you that, and that is the cuffed or comma shape bacteria. If you are understanding that that is a comma like shape, yes or no, right? It's a comma shape bacteria. So that whenever we have understood about the basic bacteriology classes, we have already lots of time listened that the Vibrio cholerae is the comma shape bacteria. Comma shape, got it? Comma shape. After that, see here, that basically causes the cholera, one of the severe diarrheal disease into our surroundings. Cholera was most prominent into the early era. Nowadays it's completely eradicated, or nowadays the prominence of the particular disease is less, but into the previous era cholera was one type of pandemic situation also. Cholera, got it? See, first described, see here, Pacini 1854. After that, confirmed by the Koch. See here, 1883. Found where basically founded? See here, Brackish and the salt water. Basically, they are founded the cholera. It is also one type of waterborne disease, that is the cholera. See, after that, it is facultative anaerobic and highly motile. Highly motile, defining that they can easily move because they have the flagella. And facultative anaerobic, it basically defining that they cannot grow without oxygen, but they can also grow with oxygen. It defining that the facultative term always defining that without these particular things they can also grow. Facultative defining that. Okay, now I'm coming to the second slide. See here, we are coming to the morphology. We need to know how the morphology of the Vibrio cholerae. See, it is the comma shape bacteria, got it? And here is the flagella, so that they can highly motile. See, shape, firstly we need to understand that is a comma shape. I have already told you that comma rod shape. See here, that is a rod shape and that is a comma like shape. See, second one is the size 2 to 4 micrometer, length, and the width, that is 0.5 to 0.8 micrometer. Got it? See, after that, motility, single polar flagellum, so that they basically motile as well as their motility is appearance is darting motility. Got it? It is also important for your competitive aspect also. There have lots of question might be occurred from this darting motility. Which bacteria defining or which bacteria express the darting motility, you have to say that that is the Vibrio cholerae. Got it? See here, here is a flagella, so that it's basically darting motility we can easily get. Got it? See, after that, that is a gram stained pink in color. Why? Because that is a gram negative bacteria. See, here is a non-sporing and may have capsule. Whenever the capsule are founded, they basically too much virulent. Capsule basically enhance the virulency of the particular bacteria. Okay, I have already told you that how to you understand the morphology. I am again saying you that you need to memorize, firstly, is it gram positive or gram negative? first. Secondly, you need to understand that is it how is the morphology, is it comma, is it cocky, is it bacilli, you need to understand it. Thirdly, you need to understand it, is it aerobic or anaerobic. If aerobic, which kinds of aerobic, strictly aerobus or facultative aerobus. Fourth things you need to understand that is it motile, and if they are motile, which kinds of flagella are present. Fourth thing you need to understand. Fifth things you need to understand, is it capsulated or non-capsulated, you need to understand. Six things you need to understand, that they are spore forming or not spore forming, you need to understand it. Got it? See, these kinds of things or these kinds of five things you need to understand whenever you are discussing the morphology of any kinds of bacteria. I am again saying you that I always takes the class, I always try to express myself in the way that you can take it for your entire life, not only just memorize, but also you need to take the concept for your entire life, so that you cannot forget the particular things. See, now we are coming to say that is the cultural characteristics. We have already understood the morphology. Now we are coming to the culture characteristics. See, firstly, growth. The culture defining that the particular bacteria grow, where they are growing, it defining the culture, right? See here, that is the medium that should be alkaline. Got it? That is a 8 to 9.5 that their pH should be, because the pH is more than 7, it defining the alkaline. Got it? See, after that, that is a halophilic. After that, media, that is the TCBS agar we may uses TCBS. Please keep in mind that TCBS agar. After that, APW, it is also one type of agar, it is also one type of enrichment agar, so that they are here to growing the Vibrio cholerae and inhibiting the another type of bacteria. Got it? They basically help to grow the desired bacteria and inhibit the another bacteria. Got it? After that, into the blood agar, sometimes they got the beta hemolysis. Beta hemolysis, they are red in color. Got it? After that, see here, pink agar, they are non-lactose fermentation happening, or they are not fermenting the lactose, so that they basically defining the colorless. Got it? Colorless. If it's lactose fermentation are happening, then the color become pink. Yes or no? Then the color become pink. Got it? See, and it is the TCBS agar. Got it? Within the TCBS agar, they basically forming the yellow colonies. If you are noticing here, it is also yellow colony. It is also yellow colony. It is also yellow colony, and they are have also one important thing for your competitive exam purpose. Please keep in mind that there have lots of time. It's the askable question that. It's the askable question. See here, it's the askable question that which kinds of agar or within the TCBS agar, which bacteria we can grow, then you have to say that Vibrio cholerae. Got it? It is also important things, you need to memorize it. After that, now I am coming to say here. See here, I have to choose the another color of the particular pen. See, we have already understood the cultural characteristics. Now we are coming to the biochemical test. Which kinds of biochemical test is positive? We just need to take the idea. First one is the oxidase, it's positive in nature. After that, we are coming to the catalase, it's also positive in nature. After that, we have to do the indole, it is also positive. Into the Escherichia coli, yesterday's class I have already told you that indole Escherichia coli is also indole positive. After that, stirring test, it's also positive. After that, they basically ferment glucose fermentation, they basically ferment sucrose, they basically ferment mannose, but they are not producing any kinds of gas. Got it? After that, that does not ferment the lactose. Please keep in mind that here is also an important question that Vibrio cholerae ferment, there have lots of option. Then if you will get the lactose, then you have to choose the another one. They are non-lactose fermentation, as well as they are non-lactose ferment bacteria. Got it? After that, they basically reduce the nitrate to nitrite. Got it? Nitrate to nitrite. Understood? Because that is a gram negative bacteria, so that they basically nitrate reduction are happening. These kinds of biochemical tests might be occurred, as well as we may do for identification of the Vibrio cholerae. Got it? See, now we are coming to the antigenic characteristics. Please keep in mind that it is the gram negative bacteria, so that there should be present lipopolysaccharide LPS. Okay, and I have already told you that there have lots of antigenic type, right? O antigen defining the somatic antigen. I have already told you that. See, there is a more than 200 serotype basically present, or whenever we are defining or we are classifying the antigen based on the bacteria, they basically called the serotype. Got it? So that there should be more than 20 serotype. After that, see here, epidemic strain O1 and O139. These all are important for your examination purpose. Please keep in mind that that's all are important for your examination purpose. O1 is also important. O139 is also important. They all are the epidemic strains. Got it? And here is also the same morphology. Just look down upon the particular things for your visualization, or for your understanding, or for your memorization, or for your just identifying in nature. And I have provided the picture here. See, after the O1 biotype, that have also biotype, that is a classical type, that is a EL-TOR, that is also type. And there have also the classification of the serotypes here. Inaba, after that Ogoa, after that Hikojima. These all are important. Please keep in mind that these all are important for your examination purpose. You need to just take the idea, you need to just memorize the particular name. Got it? Now I am coming to, sir, that is the age antigen. I have already told you that age antigen always the flagellar antigen, F antigen always fimbrial antigen. I have already told you that O antigen always somatic antigen, right? See, after that, see here, that is the disease condition. We need to take the disease condition, as well as we need to take the idea regarding the disease condition. Hope the particular thing is clear up to this much. Got it? See, here is a disease. Vibrio cholerae basically causes one of the prominent disease, that is the cholera. We all known about the cholera because into our surroundings cholera is too much common, right? So that here is an important question regarding the cholera. You might find out into various type of examination. See here, rice water stool. Lots of previous year question you will find out that is the rice water stool basically caused by cholera. Rice water stool basically we see into the cholera. There might be comes the questions that rice water stool is caused by, there have lots of bacteria name, you have to choose the Vibrio cholerae. Got it? And within the cholera, which kinds of symptom might be occurred? See here, rapid dehydration might be occurred, shock might be occurred, death might be occurred. Got it? See, there is a cost how the cholera might be occurred. See, that basically caused by the cholera toxin. We can say it into the short form, that is a CTX. Got it? And there might be also the condition in the too much rare condition, that is the spectemia and the wound infection. Okay, that's all are the disease condition. So that you need to just take an idea regarding the antigen and the disease condition. I have already told you that what are the important things you need to memorize. Okay, now I am coming already last stage or the ending stage of the particular lecture. There have the lab diagnosis and there have the treatment or prevention. See, lab diagnosis, how do you do? Firstly, you have to collect the specimen. You have to collect the stool swab or rectal swab. After that, you have to do the culture in TCBS agar. I have already told you that. After that, what will you do? You have to do the gram staining. You will get the gram negative rod or Vibrio or coma like shape. See here, it is also one type of microbiology microscopical structure. It is also one type of microscopical structure. You just take the very basic idea of the particular bacteria. Got it? After that, I have already told you that the motility, that is a darting motility, you will get. Understood? After that culture, I have already told you that APW, TCBS agar, you will get the yellow colony. After that, the biochemical test, I have already told you that E oxidase positive, indole positive, stearin positive. Got it? After that, whenever you are just doing the serological test, you need to take the antigen at first, because without the antigen, serology might be occurred. So the antigen and antibody reaction basically occurred the agglutination. Got it? So you see here, that is a O1O139. These two antigen are the epidemic antigen. You will find out lots of question from here. Epidemic antigen. Got it? After that, if I am coming to the molecular test, how do you do it by the molecular biology? See here that we have to do the PCR, that is a polymerase chain reaction. Got it? And CTX gene, I have already told you that CTX basically producing the cholera toxin, and this cholera toxin basically help to causes the cholera disease. Got it? So that's all are the laboratory diagnosis. We have already covered in every aspect of the laboratory diagnosis. See, now I am coming to the last stage of the Vibrio cholerae. See, we have to firstly prevent it. How do we prevent it? See here, we have to wash our hand properly. We have to drink safe water. We have to eat proper cooked food. Okay, as well as we may take the cholera vaccine also, as well as we have to avoid the raw food. Firstly, we have to cook it properly. After that, we have to take. That's all are the cholera prevention. Okay, now I'm coming to the treatment stressors. See here, that is a rehydration, and this cholera might be also occurred by the ingestion, like contaminated food, drinks, they basically occurred. Okay, rehydration are the life saving, because whenever the cholera are happening, there have lots of dehydration are occurring, because there is a rice water stool are happening, so that lots of water are extracted out through the stool, so that we have to take the water at first. That's the terminology that is called rehydration. Got it? After that, oral rehydration, that is a ORS we have to take. After that, we have to take the IV fluid. After that, there have also some kinds of antibiotic we may use, or the doctor may suggest into the case of the cholera. See, that is a doxycycline for the adult, and I am not emphasis into the treatment part, because treatment part is not our part. We have to just diagnose it. After the part is the under the doctor basically suggests the medicine, but they have mentioned some kinds of treatment name or some kinds of antibiotic name, so that we have to take the idea, just a basic idea for our surroundings. See here, azithromycin we can also take for the children and pregnancy. After that, ciprofloxacin, after that tetracycline, they all are the alternative antibiotic. Got it? And the zinc, they basically reduce the diarrhea in the children. Whenever we are taking the zinc supplement, as well as we are providing the zinc supplement to the children, it basically helps to reduce the diarrheal condition. Okay, hope that thing is clear. So that we was starting the class from introduction, right? And we are coming to the treatment portion within a small time. Hope you will get a elaborate idea regarding the cholera. Got it? Thank you so much for attending. Just check our description box. There have also lots of free services you will get from our platform. We have our ebook, we have our competitive exam batch, we have our tuition batch, we have our own even lots of ebook. Okay, we have our own test series, and there have also lots of services are available. We always give the best level of education in the M.L.T students, okay, and in the field of medical lab technology. Okay, thank you so much for listening properly.